So Bastien Paré is at the University of Laval. He is a graduate student doing his PhD in the laboratory of Dr. Francois Gros-Louis, and is a recipient of the 2016 ALS Canada Doctoral Research Award, and working on a really cool niche in the field of ALS. So I hope you enjoy it. And Bastien, if you're there, the floor is yours.
All right. Thank you, Dr. Taylor, for the presentation. So as David said, today I'm going to talk about a part of my PhD project, which is the development and characterization of a tissue-engineered skin model to study ALS. So let me, are you seeing the Go webinar box?
Oh, we did just see it. Now I see it.
You see it?
Sorry? Oh, no, we don't see the interface, the thing at the right. No. Okay, we only see your slides. Yep.
Okay, great. Okay. So, ALS, as you might all know, was first described by Jean-Martin Charcot in 1874. It's a neurodegenerative disease affecting the motor neurons and generally causes a progressive paralysis and muscle atrophy. So the rationale behind my project is that one major obstacle for the study of ALS is the availability of neuronal samples. So we think we need a human model in order to understand the development of the disease and also to discover biomarkers which would be associated with the disease and to develop effective drugs. So for example, predictive biomarkers in ALS could help get a better diagnosis, earlier therapies for patients, as well as better clinical studies. So early diagnosis will become important in the treatment of ALS to save motor neurons before degenerescence, since we know that around 50% of them are degenerated at the diagnosis. So you might wonder, why is this tissue-engineered skin model interesting for the study of ALS? Well, because there's a link between ALS and the skin. We know there's a connection between the brain and the skin. There are the sensory receptors, as well as the fact that they both come from the same embryonic sheet, which is called the ectoderm. There's also a lot of skin changes that have been described in ALS patient skin before the neurodegenerative disease, and also in other neurodegenerative diseases before the apparition of clinical symptoms. So in ALS, we were able to detect the phenomenon called the delayed return phenomenon. There's also lower collagen fiber diameter and an increase in the immunoreactivity of some proteins as VEGF, IL-6, IGF-1, TDP-43, VCP, and ubiquitin. And changes in the skin have also been described in Parkinson's disease patient and Huntington's disease patient. So firstly, I want you to show what this I want you to see what this tissue-engineered skin looks like. So on the left side of the screen, you can see tissue-engineered skin derived from healthy patients. Those are control tissue-engineered skin. In the center, you can see TES derived from C9orf72 mutation carrier familial ALS patients. And on the right, you can see TES derived from sporadic ALS patients. And as you can see when you compare to control skin, there are a lot of structural abnormalities as epidermal differentiation, dermo-epidermal junction, delamination, keratinocytes infiltration, as well as collagen misorganization. So it seems that the skin fibroblasts from the skin of ALS patients have a problem in the secretion and assembling of extracellular matrix compared to cells which are derived from controls, so healthy patients. We wanted to go a bit further and see if ALS protein could be associated, ALS-related protein could be associated with our tissue-engineered skins. So we've checked for TDP-43. As Dr. Taylor mentioned before, TDP-43 has been associated for a long time to ALS. So the pathological model of the protein is that in normal healthy neuronal cells, TDP-43 will be mainly detected in the nucleus of the cells, as in ALS patients' motor neurons, TDP43 will mislocalize to the cytoplasm and form cytoplasmic aggregates. So we wanted to see if our tissue engineered skins derived from ALS patients, if we could see in them TDP43 cytoplasmic aggregates. So what you can see here is TDP43 mislocalization in ALS-derived tissue engineered skins. So here you can see a LT tissue engineered skin. So in blue, which is called DAPI, you can see the cell nucleus, and in green the protein TDP43. And in the LT tissue engineered skin, TDP43 is mainly detected in the nucleus of the cell, as when we check in C9 or 72 derived TES or sporadic ALS patient derived TES, TDP43 seems to mislocalize in the cytoplasm of the cell, so out of the nucleus, and form cytoplasmic aggregates. We also quantified this TDP43 mislocalization to be sure what we were seeing here is really cytoplasmic TDP43. And what we were able to see, in fact, was that in the control tissue engineered skins, so derived from LT patients, TDP43 was mainly detected in what we call the nuclear fraction, so the nucleus of the cells, and only a bit detected in the cytoplasmic fraction of the cells, so the cytoplasm. But when we check the same thing, TDP43 in the C9 or derived TES or in the sporadic ALS TES, what we can see is a shift where TDP43 comes mainly present in the cytoplasmic fraction of the cells and less present in the nuclear fraction of the cells. What's interesting is that we were not able to detect this cytoplasmic mislocalization of the protein TDP43 in the cytoplasm of C9 or 72 skin fibroblasts in 2D culture model. So here, again, you can see what we call a immunofluorescence, where the nucleus of the cell are in blue and TDP43 in green. And we were not able to detect any TDP43 in the cytoplasm of the cell, showing that our tissue engineered skin, the TES model, the 3D cell culture model, could be necessary to see TDP43 mislocalization in ALS-derived skin fibroblast cells. Finally, we wanted to check if this phenomenon was also observed in native skin, human skin biopsies. So we took normal human skin biopsies as well as sporadic ALS skin biopsies. So what you're looking at in A are normal human skin biopsies. So in brown, you see the protein TDP43, and in blue-gray, the nucleus of the cells. And what we can see is that mainly TDP43 is present in the nucleus of the cells, as when we check for in native skin of sporadic ALS patients, we can see TDP43 being mislocalized and forming aggregates or accumulation in the cytoplasm of the cell, where the nucleus is again in blue and gray. And we're also able to detect in C accumulation of TDP43 in the spinal cord of these same sporadic ALS patients. So in conclusion, we report the generation and characterization of a novel in vitro engineered skin model to study ALS. Of particular interest, cytoplasmic TDP inclusion, a well-established pathological feature of ALS, were uniquely detected, seen in our ALS-derived TDS, as well as in native skin biopsies collected from corresponding ALS patients. They were in fact detected in sporadic ALS-derived skin, which were not at the end stage of the disease, and in pre-symptomatic C9 or 72 familial ALS patient carrying the GGGGCC DNA mutation. Furthermore, our ALS-TES also presents a number of abnormalities associated with the extracellular matrix. So we think the application of tissue engineered skin models, we think we could applicate it to facilitate the identification of biomarkers for early diagnosis, as progression will become and it will become highly attractive and will be of high interest for future research, allowing monitoring of patients while the disease is getting on. And then I will briefly go on these results, but we wanted to use this tissue engineered skin model to discover novel biomarkers which could be associated to the disease, to ALS, and then maybe develop drugs against those biomarkers. So what we did is we optimized an approach to recover secreted proteins from fibroblast conditioned media for secretomic analysis to discover biomarkers. And what we thought was quite interesting with the results we've got is that there were small molecules that we thought could be interesting to study in ALS, and these molecules, or extracellular vesicles, in fact, are called exosomes. So exosomes are membranous vesicles composed of lipids presenting with molecules such as proteins, RNAs, and microRNAs. They have a diameter between 25 to 150 nanometer, and they are a possible pathway for transfer of protein, lipids, and RNAs from cell to cell. So right now, nobody has ever showed that fibroblast cells can efficiently secrete exosomes. So our hypothesis, since we were able to detect exosomes-associated proteins, is that our fibroblast cells culture in a 3D fashion could represent a great source of exosome and a potential source of ALS-related biomarkers. So first, we wanted to quantify the secretion of exosomes from fibroblast cells in a 2D culture model compared to our tissue engineered skin, the 3D cell culture model. So we've quantified exosome protein, and we've quantified it from exosomes from skin fibroblasts and also bladder fibroblasts, just to be sure that all kinds of exosomes of fibroblasts would secrete exosome. What is very interesting for the study of ALS is that in fibroblasts collected from the skin of patients, who are able to detect around 30 more time the amount of exosome protein derived from our 3D cell culture model, the TES, compared to the TD cell culture model. We also wanted to see if this exosome secretion could be modulated. So we've added to the cell a compound which is called GW4869, which is a known inhibitor of exosome secretion. And what we're able to see here, in fact, is that when you add this compound to fibroblast cells, you can inhibit the secretion of exosomes. And as exosomes could be implied in the pathology of ALS, we think that their secretion modulation could be interesting in the biomarkers and drugs aspect of the research, but I'll get back to this a bit later. And we wanted to see what those exosomes derived from skin fibroblasts look like, because we were kind of the first to be able to get them in a high concentration. So we checked them via DTEM, which is a transmission electron microscopy. And exosomes, in fact, are small vesicles of around 100 nanometer that you can see here in gray or white. And since exosomes are known to be able to transport proteins and RNAs from cell to cell, we wanted to see if the collected exosomes we've got from fibroblast cells could enter and penetrate cells as they should really do. So this is an experiment we did. So here you've got fibroblast cells for which the nucleus are stained in blue, and exosomes have been stained in red, and we've added exosomes stained in red in the cell in the culture supernatant of skin fibroblasts. And here, after 48 hours, you can see the apparition of the red color in the cells, showing that the exosomes we are collecting can enter fibroblast cells, so they can really transport proteins and RNAs from one cell to another. So going back to ALS, we wanted to see if the exosomes derived from skin fibroblasts, which would be collected from biopsies from sporadic ALS patients, C9orf72 or SOD1 mutation carriers, could contain ALS-related protein. We first checked for the SOD1 protein in our exosomes. So SOD1 is a copper-zinc superoxide dismutase, which is abundant and ubiquitously expressed. It accounts for around 15% of familial ALS forms and around 1 to 2% of all ALS cases. There's no special clinical features associated with SOD1. There's more than 142 different missense mutations identified in 74 different codons, which represent around 50% of the protein, which is kind of big. Some mutations we know will affect survival time, as the A4V1 mutation that I will talk a bit later about, and some affect the disease onset. So first we wanted to see if in exosomes derived from fibroblast cells from sporadic ALS patients and A4V patients, if we're able to detect the toxic SOD1, which is called misfolded SOD1. So we did an analysis that is called a Western blot, and the presence of the protein can be associated by the apparition of this small black band here. So we've checked for 2 species of misfolded SOD1 that are called B8H10 and C4F6. And in the exosomes derived from these fibroblast cells, we were able to detect those 2 strains of SOD1, misfolded SOD1, in fact, and in exosome derived from sporadic ALS patients, as well as exosome derived from A4V mutation carrier, which are also ALS patients. And finally, since we now know that misfolded SOD1, which is a toxic protein associated with the development of some ALS cases, is present in our exosomes, and since, as I showed you a bit earlier, we know that the exosomes derived from skin fibroblast cells can go from one cell to another, we wanted to see if misfolded SOD1 was able to go from one, let's say, sick cell, so ALS-related cell, to an LT cell, and then get into the LT cell. So what we did is that we took skin fibroblasts from LT patients, and we cultured them in the lab. So on the left panel, you can see LT skin fibroblast cells for which there was no addition of exosome in the culture media or anything, and we stained the nucleus of the cell in blue, and we stained for the protein TDP43 in red, and we also stained for the C4F6 misfolded SOD1 species in green here. And as you can see, LT patients' skin fibroblasts don't show any misfolded SOD1 and not any C4F6 SOD1, which could be detected by seeing green aggregates or small green points. But when we take those same fibroblast cells and we add A4V-derived exosomes after 48 hours of culture, we can detect misfolded SOD1, the C4F6 species of misfolded SOD1, in our fibroblast cells, which are LT fibroblast cells, which tend to show that these exosomes can help toxic associated protein as misfolded SOD1 go from one cell to another, and go from maybe one cell that do secrete misfolded SOD1 to a cell that wouldn't contain any misfolded SOD1 or wouldn't secrete any misfolded SOD1 at all. So in conclusion, our 3D cell culture of fibroblast cells enhance exosome secretion when compared to the TD cell culture, which shows that the tissue engineered skin could be interesting in the discovery of biomarkers, which could be inside the exosomes. These exosomes have the capacity of entering fibroblast cells, therefore allowing transport of lipids, protein, and RNA. Here I showed only transport of misfolded SOD1 from one cell to another, but there are RNAs, other proteins, and lipids that could be interesting to study and that could go from one cell to another. Misfolded SOD1, a protein associated with the development of some cases of ALS, was found in fibroblast secreted exosome in a culture model from sporadic ALS and A4V patients. And skin fibroblast derived exosomes seem to allow the transport of molecules as protein from cell to cell. So quickly, I'd like to thank Dr. François Gros-Louis, which is my PI, our collaborators here in Quebec, Nicolas Dupré, and in Montreal, Guy Rouleau and Patrick Dion, also every member of Dr. Gros-Louis' lab, and the funding agencies. So that wraps it up.
Thank you, Bastien. That was really great. I think it's, as I said, a really interesting niche, and it's kind of cool because I think so many of the studies that were done before on skin just weren't very well powered, and now having you focusing on that, hopefully we'll be able to see what can really come out of that, and especially with the new data that you're showing, is pretty cool stuff. We do have a couple questions, but I'm going to just give you one from one of the listeners, which is that why is the mislocalized TDP43 diffuse in some of the SALS patients and more punctate aggregates in others?
Well, this is not something we've studied in depth, but probably we know that C9 or 72 aggregates in the tissue and general skin are more very punctuate TDP43 aggregates, and in sporadic ALS patients derived TES, it's more like a diffuse accumulation. Unfortunately, we don't know why. Is it because of the mutation of C9 or 72 in the patients for which we have skin fibroblast cells in the lab? Is it the way they were extracted? It's hard to say why the staining of TDP43 is different. What we thought was interesting is that TDP43 mislocalized in the skin of ALS patients, which is out of the central nervous system, and we think it's the first time someone has ever showed that TDP43 mislocalization can be detected outside of the CNS. So, but why we see different staining, more studies should be done about it. But since all our patients were not at the same stage in the symptoms as the C9 or 72 patients were pre-symptomatic when the skin biopsy was took, maybe there's a play in the time of accumulation of TDP43 or something like that.
Okay. Well, thank you very much, Bastien. We have some other questions, but maybe we'll forward those to you afterwards, and we really appreciate your time and wish you a wonderful rest of the day.
Well, thank you very much.
Thank you.